Driving pressure is the pressure that moves substances from the blood plasma into the nephron. And that's what we're going to talk about in this video. Just a quick review. In green right here is Bowman's capsule. Leaving Bowman's capsule is the proximal convoluted tubule. So this space right here, this open space in white, is the capsular space. That is where filtrate is first going to accumulate. Then it's going to proceed down the proximal convoluted tubule, and then throughout the loop of Henle, distal convoluted tubule, and the collecting duct, and then be excreted into other regions of the kidney. We also have the capillary bed right here, known as the glomerulus. Bringing blood to the glomerulus is the afferent arteriole, and taking blood away from the glomerulus is the efferent arteriole. So like we've talked about in capillary beds and other regions of the body, almost everything in our blood moves from the capillary bed into the tissues. Now, we're not talking about movement from the capillary bed into the tissues. We're actually talking about movement from the capillary bed, from the glomerulus into Bowman's capsule, or specifically into the capsular space. And what leaves the blood is nearly everything, with the exception of the formed elements, white blood cells, red blood cells, and platelets, and the large plasma proteins. Some smaller proteins can certainly get filtered, but what gets filtered is ions, minerals, vitamins, biomolecules, fats, lipids, amino acids, small proteins. It's a great way to get rid of hormones from the body, and certainly carbohydrates like glucose. So nearly everything other than the large plasma proteins and the formed elements. And the driving pressure is what ultimately determines the net movement of substances from the blood plasma into the nephron. So let's take a look at the pressures. And there's a number of pressures that factor in to this. One of it is the pressure in the glomerulus, or the glomerular capillary pressure, which I'm just referring to as blood pressure because that's ultimately what it is. Traditionally, at capillaries throughout the body, capillary pressure is around 35 millimeters of mercury. It's a bit higher in the glomeruli of the nephrons, and that is going to push substances from the plasma into this capsular space right here. Just so we're clear, the nephron is lined with cells, specifically epithelial cells, and the type of cells lining the Bowman's capsule are simple squamous epithelial cells. So we see that here, and then on the outside as well. This is known as the visceral layer. Out here is the parietal layer, but between the visceral layer and parietal layer of Bowman's capsule is the capsular space, and that is where filtrate moves to. But the blood pressure within the glomerulus is opposed by two other pressures. One of them is the colloidosmotic pressure. And once again, this is no different than any other capillaries we've looked at throughout the body. What we talked about in previous videos is the colloidosmotic pressure that is created as fluid leaves the capillaries. As fluid leaves the capillaries, but those large plasma proteins remain in the capillary bed, that creates a high solute concentration in this capillary bed, in this case the glomerulus, and that high solute concentration is going to attempt to draw water back into that capillary bed, back into the glomerulus. And this stands for colloidosmotic pressure. That's pressure within that's the colloidosmotic pressure due to the plasma proteins in the glomerulus. And that is roughly 30 millimeters. There is another pressure opposing blood pressure, and that is capsular pressure. So once again, this is Bowman's capsule. And in here, filtrate starts accumulating. Within the filtrate is a lot of fluid or water. And any time fluid or water is within a confined space or container, pressure is exerted on the walls of that container. And that is no difference in the Bowman's capsule I should say it's no different in Bowman's capsule than it is in the glomerulus. So there's pressure that is created as fluid starts accumulating in this capsular space, and that pressure is about 15 millimeters of mercury. So if we look at all three of these pressures, the glomerular blood pressure is 55 millimeters of mercury, colloidosmotic pressure roughly 30, and the capsular pressure at 15 millimeters of mercury. So colloidosmotic pressure and capsular pressure are opposing blood pressure. So our body needs to maintain blood pressure at sufficient levels so it can overcome these other two pressures. And that's what we see here. And this is how we get the net driving pressure of 10 millimeters of mercury. Now, one more thing I want to talk about is what prevents these plasma proteins from moving from the glomerulus into Bowman's capsule. One is the size of the plasma proteins. They are too big to squeak through these perforations in this capillary bed. But two, within this blue area, which is the basement membrane, there's some negatively charged glycoproteins, some molecules that have a negative charge. Because plasma proteins are also negative charge, that negative charge within the basement membrane repels the negatively charged plasma proteins. So even if there are some plasma proteins that are small enough to squeak through these fenestrations, they're almost certainly going to be repelled due to the negative charge within this region. So this is net driving pressure. We'll talk about the very importance of maintaining that and how that is regulated.